of environment we receive with the eyeball. So the eyeball is round in shape. You can compare it with the globe. It has two poles. Pole is anterior, pole is posterior. Farthest points of the eyeball. It has an equator in this place. So if you connect the two poles, it will be the line named anatomical axis. We have external and internal anatomical axis. Internal axis connect internal points. External axis connect external points. I'll continue after the break. So, the third axis. The axis which connect the center of the lens with the place of the best vision. The place of the best vision, named macula. Forward centralis. Macula forward centralis. If you look at the small point, it's projected on this place. So, this axis has name axis opticus. Bon appetit. Axis opticus. And axis anatomicus, which connect various points of the eyeball. So it consists of three layers: external layer, tunica fibrosa, the fibrous layer. Two places. There are two parts on this layer: cornea and sclera. Cornea seems like a watch glasses. It's transparent. It has no vessels, has no nerves. So that's why the light passes through it easy. Sclera, white in color. It's larger at the posterior side. If you compare the size of sclera on this place, as you see, less larger here. Amos. Repeat after me. No?
Sorry.
Sorry, are you Amos?
No.
No. Not him. Speaks everything himself. So, between sclera and cornea, in this place, you can see sinus venosus sclera. It's around the margin of cornea. Locates inside. It is a canal which receives the fluid from the chambers of the eyeball. This fluid enters to this canal and moves out the eyeball. In case the passage of this fluid is blocked, the pressure inside the eyeball is enlarged. Glaucoma, the name of this disease. So, next layer. Tunica vasculosa bulbi. Three parts. Choroida. Choroida consists of vessels which supply the eyeball inside. Its continuation, corpus ciliaris, and last part, the iris. All the structures are same vasculose layer, but they have different function. Corpus ciliaris, triangular in shape here, consists of smooth muscles. That muscles have three directions: meridional direction, radial direction, and circular direction. Connected with the ligaments of the lens. Contraction of those muscles help to change the shape of the lens. In case you look far away, the lens is flat. If you look things near to your eyeball, it is round by contraction of muscles. So, another function of the ciliary body, it has processus ciliaris which produces the fluid Humoracosum, you know that it is a fluid which must be absorbed to sinus venosa sclera. So, this fluid travels from processus ciliaris to the posterior chamber and then to anterior chambers. The chambers separated one from another with the next part of tunica musculosa bulbi, with iris. You see? This one is a disc with the hole inside. The name of this hole is pupilla. The size of this pupilla may be changed correspondingly to the lightness. Much light, less size of the pupilla. Less light, the large size of pupilla. It is regulated with two muscles. Musculus sphincter pupilla in the circular direction and musculus dilatator pupilla with the radial direction. That muscles innervated with parasympathetic and sympathetic system correspondingly. Parasympathetic system with nervus oculomotoris innervates musculus sphincter pupilla. Parasympathetic system innervates musculus dilatator pupilla. In darkness, musculus dilatator pupilla enlarge the pupilla. Now, if you come to the street, you will see that your pupil will decreases in size by musculus sphincter pupilla. So, next, the color of eyeball depends on pigments in the iris. In case little pigment, blue eyes. More pigments, green eyes. Much pigments, black eyes. And so on. So next, tunica interna is retina. Retina consists of two parts, pars pigmentosa and pars nervosa. Actually, there are two layers. External layer is pars pigmentosa. This one, external layer. Pars pigmentosa consists of pigmental cells. Pars nervosa consists of neural cells. The direction of light is here. So pars nervosa is transparent. The light moves easy and irritate receptors. Receptors located in bulbs and cones. Rods and cones, sorry. Rods and cones. They feel the light and colors. Rods work in darkness and they see the gray color on the cones, are able to differentiate red, blue, green and so on, all colors. But they don't function in darkness. The most place of collection of cones, this one collection, over centralis. In this place, no rods, only cones. That's why we can see in lightness by this point. In darkness, we cannot see with it. In darkness, we cannot differentiate color. If you see the car, you cannot understand is it green or green, or red, and so on. So, three layers of cells. The first, rods and cones. The second layer, bipolar cells. Bipolar cells. The next layer, ganglionar cells. All visual information moves from rods and cones to bipolar cells to ganglionar cells, which axons collected and go out the eyeball. The collection of the axons is here. It will be nervus opticus. It moves through discus nervi optici. And you understand, in this place we have no rods and cones. That's why we cannot see with that place. It is a brief point. So, then, axons from ganglional cells, go to the next cells. That says cells present in corpus geniculatum laterale. What is corpus geniculatum laterale? You see the metathalamus laterale from superior colliculus of rhomboid fossa, of mesencephalus midbrain. So, from corpus geniculatum laterale, the axons go through capsula interna. You remember the capsula interna is here, and posterior side of capsula interna is posterior place is radiatio optica. So radiatio optica is collection of fibers which go from corpus geniculatum laterale. Finally, they go to medial surface of the occipital lobe of the brain. But look, axons of ganglional cells decussated partly in this place, chiasma opticum. You remember that it is hypothalamus. Look at it. Only medial portions are decussated. You see? Lateral portions continue to move through the same half of the brain. And finally, each unispheres of the brain receive two halves from both eyes. The left occipital lobe receive information from the left half of the left eye and the left half of the right eye. You see the blue color of this pathway. The right lobe receives the right half of the right eyeball and the right half of the left eyeball. What does it mean? It means that in case of destruction of this pathway, you can understand what place of destruction. For example, if they destroyed the nervus opticus, the information will be blocked from the corresponding eyeball. In case of destruction of tractus opticus, corpus geniculatum laterale, capsula interna, or corresponding place of the cortex, will be blocked information from two halves of both eyeballs. And the patient will see only half information. And we can examine it by following way. Find the middle place. Yes. In case the patient had a problem with the eyeball, he sees only he will see only this half. He will find this point. Understand? So, next question. Next question, refractory medius of the eyeball. The light moves through the eyeball by following structures. First, cornea. Second, humor aquosus, which present in camera bulbi anterior and camera bulbi posterior. Next, lens. The lens consists of capsular lenses, which connected with the zona laterales, ligaments which attach to the cellular body, and nucleus lenses inside. Next, corpus yellow like a jelly in back of the left side. It is the largest refractory media of the eyeball. It has no vessels, no nerves. All the structures have no vessels, no nerves. They're transparent. So, for the next class, structure of the eyeball and pathway of the visual analyzer without pathway of pupillary reflection. Only visual analyzer.